Welcome everyone. For the first presentation, we will welcome Corentin Bernard, who will give a presentation about data-driven sampling strategies for fine-tuning bird detection models.
Thank you. So, hello everyone, I'm Corentin Bernard. I'm doing a postdoc at Toulon University in France. And I'm going to present my work on data-driven sampling strategies for fine-tuning bird detection models. So my work is part of the TabMan project, a European project with partners in Spain, in the Netherlands and in Norway. And our goal is to deploy a transnational acoustic monitoring network with the same protocol and the same material in every country to study bird migration. As recorders, we use microphones like this with solar panel and internet connection to continuously record the acoustic environment and be able to detect any bird that would spend a few minutes nearby. In the project, my work focuses on the automatic detection of bird species. In the literature, there are already some pre-trained models, such as BirdNET, but these models can be affected by various factors, like the species distribution that we have in our sites, the acoustic environment, the recording material, and the bird regional dialects. However, these models can be improved through fine-tuning with annotated in situ data. So that's what we want to do, but we have a problem. Passive acoustic monitoring collects a huge amount of data, and bird vocalization annotation requires a specialized expertise. As a result, only a few portions of the recordings can be annotated. And this kind of data is also very sparse. Only a small portion of the recordings contains birds. And there is a high class imbalance between rare and abundant species. So we have a question, is how to select the most relevant samples to be annotated in order to improve the model. In this study, we use the WABAT dataset that contains soundscapes recordings with annotated bird vocalizations, to which we added another dataset of environmental noise to simulate low bird call occurrence that we have in passive acoustic monitoring. And we assume the following scenario. So we cut the recordings into 3-second splits. That's what I'm going to call as a sample. And so that I have a dataset of 300,000 samples. And we assume that we have the limited annotation budget with a maximum of 500 samples that can be annotated. So the question becomes, what is the best strategy for selecting the 500 samples to be annotated in order to maximize the model performance? As a model, we use the BirdNET to compute the embedding, and we fine-tune BirdNET by training the last layer here. The first part of my work investigates how to know which samples are valuable for training. To do so, we use the methodology called reverse correlation. So here is my train set, and if I select totally randomly 500 samples, I can train a model, so fine-tune BirdNET, and then evaluate this model to get a performance metric. As metric, we choose the mean average precision, and the higher, the better. We can do the random sampling many times with different seeds. In this study, we did 10,000 times. And after that, we can look at one samples and compare its presence or absence in the train set with the mean average precision. And if we do the correlation between the two, we can get what is called the influence score. And we can compute the influence score for every sample in the train set. A sample with a high influence score leads to better model performance when included in the train set. This information is very valuable. We can, for example, select the 500 samples that have the highest influence score to have an idea of the best performance that we could get with 500 samples as a baseline. But to do this methodology, it requires all the data of the dataset to be annotated, which is not possible to do with new data. So the second part of my work investigates which other measurable parameters can be used as proxies for the influence score. We investigated two kinds of sampling parameters. So first, parameter based on the properties of the acoustic signal. the acoustic indices, and then parameter based on the prediction of the pre-trained models BirdNET, the maximum confidence score, and the binary entropy, a value that reflects the uncertainty of the model. With the acoustics indices, there are plenty of them, and some of them are highly correlated. So the first thing was to select a limited set of acoustic, limited number of acoustic indices. So we first looked at the distances between acoustic indices to create groups of acoustic indices that are similar. And after, within each group, we selected the index with the highest correlation with the influence score. So that way, we selected 6 acoustic indices that are clearly distinct and that are good proxies for the influence score. We can now use these acoustic indices as sampling parameters. So now we are not going to take the 500 samples totally randomly, but we are going to take samples with the parameter, so for example, an acoustic indices above a threshold. And again, we can train the model and evaluate its performances. We can do this for many threshold values to see how it evolves. And here is an example with the NDSI, an acoustic index. So first, as baselines, here in dashed line, you can see the performance obtained with the pre-trained model, so BirdNET without any fine-tuning. Here, the performance of BirdNET fine-tuned with 500 samples taken totally randomly. And here, the performance obtained with 500 samples that have the highest influence score. And here in red, you can see how the performance evolves when we take 500 samples that have a NDSI above a threshold. And we can see that the performance increases here until it reaches a peak. That's the best sampling strategy that we could get with the NDSI. And you can also notice that taking samples that have a NDSI below a threshold is worse than random. So here's a quick overview of the results that we could get with the other acoustic indices. We can see that 2 of them, the NDSI and the TFSD, works better than the others. And here are the results with the model-based parameters, so the maximum confidence score of the pre-trained model BirdNET and the binary entropy, which is the uncertainty of the model. And now if you want to compare the performances between strategies, we can just look at the best strategies for each parameter. And that's the main results. So the first thing that we see is that when fine-tuning the model, we always get better results than the pre-trained model BirdNET without fine-tuning. And the best sampling strategies was obtained with by taking samples with a maximum confidence score below a small threshold value. But the other strategy may still be interesting. That's what I'm going to investigate in future work. I want to know if some strategies are better to increase the precision of the model or the recall, and if some strategies are better with some species. I will also explore other strategies like stratified by class or based on the embeddings. And the main objective of this work is to use it as a starting point for an active learning pipeline applied to the recordings in Tommon. It means that each month we are going to annotate 500 samples and improve the model. And so the model performance will improve month after month. So thank you for your attention, and I will be happy to answer your questions.
Thank you, Corentin, for your nice presentation. Are there any questions? Yes.
Thank you so much. It was super interesting. Do you have any good practices when you have to detect different species? For example, I work with invasive species, but I also want to compare between an invader and a native species. And I found that if I don't use the same strategy for detecting them with BirdNET, it actually works better for me. Because, for example, I want a better recall with my invasive species, but for the other species, I want more precision. And I don't know, like, what would be your advice in this situation?
Good question. I would say that so far I found that some strategies are better recall and other better precision, but it may be very dependent on your data. So I would say that the best thing is to try. And if you are interested to apply this methodology, we can talk about it.
Yeah, sounds lovely. Thank you.
Any other questions?
Thank you. Hi. I wanted to ask, did you test something like the SNR? Because in example, you talk about the NDSI. NDSI compare low frequencies with high frequencies. So I think that when you select the higher scores, you select the sound with less light pollution or less low frequency. So you select the purest sound, and maybe you will miss some low frequency bird example. And so did you test something like noise ratio, something like that?
Actually, I tested them, but yes, there are plenty of them. So I had to do a selection, and I think the SNR. So I used one value, so the SNR with the frequency, and it was not so good, I would say. But I think that mainly the NDSI, like it is, it works quite well because it's able to detect birds across noise, and so yes, it's a bit. I don't exactly understand why some acoustic indices are working better than others in this work. But yes, that's what I call this work like data driven. Like I compute a lot of data and I see what happens.
Okay, thank you.
Any other question?
Yeah, thank you very much. Just about NDSI, I saw that you used the one from Scikit-Mad. I don't know if you used the native version of Scikit-Mad's NDSI, but it's very different from the definition. Like, because like it's not selecting the same spectral bands. So I don't know if you watched that. Well, anyway, if it works, that's anyway fine, I guess. But I think it would be very interesting that you might have a look at the indices you're looking at.
Yes, I use the default values of the library, but yes, a lot of them have default parameters that we could also change, and yes, it would of course change a bit the result.
Yeah, sure. Like there is many indices also like taking spectrograms values, and if you don't have a look at the window parameters, for example, of those spectrograms, that might make a big change.
Yes.
Do we have the time for one other question? Yes.
Thank you. Thank you for that talk. It was so interesting. So I guess because these are ways to select the best recordings to train BirdNET, do you think there's also scope to use that as a filtering mechanism for BirdNET output? So for example, if you process your recordings and you find that the ones with the highest NDSI are the ones that are like best informing BirdNET, maybe you could filter out the recordings that have a lower one as like some way of working with the output that's not just based on the like standalone confidence score.
Yes, maybe, but maybe I would maybe rather like use this as a filter before applying BirdNET to save a bit of computing time. But if you like run BirdNET anyway, I think it's important. It's valuable to have all the information, and after that, like filtering a BirdNET prediction with the acoustic indices seems a bit tricky, I think, but yes, maybe it could be.
Okay, thank you everyone.
Thank you.
Thank you, Corentin. And now for the next presentation, please welcome Maud Biscarat.
Thank you everyone and welcome to my talk. So I'm going to present my master thesis today that was talking about the directional regions of interest number in soundscape analysis. So it's in the following context. You may all use passive acoustic monitoring to record and monitor the biodiversity. It's a very non-invasive method, very good method, that is implying monophonic microphone, so basic microphone. And with them we record, as Corentin said, a large amount of data. And we can characterize it by acoustic indices such as acoustic richness, acoustic diversity, or the number of regions of interest that he also talked about. And in monophonic recording, we just lack the localization of sound sources. So we wanted in my internship to make a quick swap to not take a monophonic microphone, but a spherical microphone array. So basically that's a sphere with that's a sphere with a lot of microphone with it. And by pairing with a beamforming technique, we can have access to the localization of the sound sources, for example. So how beamforming technique work? Well, we wanted to find a directional bioacoustic index for soundscape analysis. So we simulated or captured the sound pressure at each microphone of a spherical microphone array. Then we projected on the spherical harmonic basis, and it's just a basis on the sphere that allows us to have a pattern of directivity, this type of pattern. And it's like if you would extract signals in every direction just with recording from a spherical microphone array. So it's really interesting because it's just as if you had a Canon microphone and you would point in a direction and extract the record a signal, but with our sphere it does it in every direction. So basically you don't have to point in a direction. And when you have the signals in every direction, you can calculate a lot of very interesting stuff, such as bioacoustic indices, and then you can create a map to have a better analysis of your soundscape than just in a monophonic recording with a passive acoustic monitoring, for example. So the bioacoustic index that we chose was the NRI. Why did we choose this one? Because we wanted to focus on one species, and the NRI is based on the acoustic signature of a species. So the acoustic signature is the duration of, for example, a vocalization, and the frequency range of a vocalization, so of a QI of a bird. And we have our signals extracted in every direction, and for each signal we are gonna compute the NRI. So for each signal we will find all acoustic events matching the region of interest wanted. So for example, for a nightingale it will be between 4,000 and 8,000 Hertz, and the duration of 0.1 second. And we count them. So in this example, there are 7 vocalizations, so NRI will be 7. To test that, we had 3 steps. First, we simulated in free field soundscape with a nightingale and a stream. So it's not random sources. One is the biophonic sources and the other is the geophonic sources, because we wanted only to localize the biophonic source. Then we reconstructed the sound field with a speaker array and captured it by a spherical microphone array. We might not have time to see that today, but we'll see. Then we did one measurement in realistic conditions to test if it was working in realistic conditions. So when we simulated a soundscape with a nightingale and a stream, so the nightingale is the red cross and the stream is the green triangle, and we computed the region of interest for each direction, for each signal in each direction. we found that the NRI was higher in the direction of the nightingale and lower in the direction of the stream. And when we compute the sound pressure level map, we can see that the stream does make sound, but it's not the sound that is searched in our NRI process, because we are spotting a vocalization of a nightingale. So that's why the NRI is quite low in this region. So it's a good way to localize only the biophonic source. But it's not the only interest of this technique. If we record a soundscape with a stream that is 2 times louder than the nightingale, we still get the information that we have a nightingale here. So that's the first step. And if I was to record with a passive acoustic monitoring, simple microphone, a nightingale, I will have this signal. So amazing, I have no other sound sources. So if I compute the NRI, I will find 34, because if you have counted, it's 34 vocalization in the signal. However, if I put another source, so a stream, you cannot really hear the nightingale anymore. So if I compute, after recording with a monophonic microphone, the NRI, I will find only 20, because the stream partially masks the nightingale. However, if I extract, as I said before, just in the direction of the nightingale, it will be a directive listening. So this is what we will hear in the same soundscape. We do not really hear the stream anymore. So it's a way for you to have a better recording. And if the nightingale is 2 times louder, you cannot hear it clearly. So if I compute the NRI, I will find only 4. So we would have completely gone by the nightingale with a simple passive acoustic monitoring. But ultimately, if I again just compute the signal in the direction of the nightingale, when the stream is 2 times louder, I have this. So again, you can a bit hear the stream, but we will hear the nightingale, and the NRI will be 29. So we will find again this information. So a directional NRI provides a good localization and is reliable no matter the signal clarity. So very useful for you bioacousticians that does a lot of field work and record sometimes not very sometimes dirty signals with a lot of pollution on top. So I think I won't have time for this, sorry. So I'll just move on to measurements in realistic conditions. So we wanted to test our region of interest maps in realistic conditions. I had to see where my birds were. So it's in a pet store, sorry, so it's not very, very realistic. But I wanted to find and to localize the canary. So we computed the NRI for canary vocalization between 2000 and 6000 Hertz for a duration of 0.1 second. They were in the middle cage. And as you can see, the NRI is higher in direction of this cage. We cannot see a big RI in this direction or in my direction. And we had quite a good map because we were in a noisy environment, because there was other sources that were masking in other direction the canaries. That's why we would find 0 region of interest in some places, because there were other sound sources such as water pumps, rain on the roof, or other birds such as parrots. But still, it doesn't provide a distinction between individuals, because we had 4 or 5 canaries in the cage, and there is still a big patch in direction of the cage. So we found that it is possible to use a directional bioacoustic indice for soundscape analysis, the number of regions of interest, and the best result, the better decay, are obtained in a noisy, polluted environment. So it's good for hybrid environment or soundscape of cities. And there is a good localization in nearby field, but there are still work to be done, especially with improving our beamforming technique to be able to distinguish individuals, especially small individuals such as birds. And we could use super resolution technique, and we would like to use arrays of spherical microphone array. So basically a few spherical microphone array, and to just have a better localization with using triangulation method to have the distance. And our ultimate goal would be to be able to census species that we will try in the Pila Natural Park in August 2027 for their census organized by the Ligue Protectrice des Oiseaux. Thank you for your listening. Do you have any questions?
Thank you, Maud. What a nice presentation.
Thank you.
Do you have any question for Maud? Yes.
Yeah, thank you very much. It's a very interesting tool. I was just wondering, what's the cost of a spherical microphone array?
Sorry?
What is the cost of a spherical microphone array?
Only 1,000 euros. So if you have a few passive acoustic monitoring and you do triangulation method, maybe it will finally give you the same cost. So that's why it's quite interesting to replace it with a spherical microphone array, except that for the moment we do not have a spherical microphone array that you can put in nature and that will last 5 years without charging it or anything. It's not developed yet, but it could be an idea.
Thank you.
Any other question? What advices should you give us to implement your directional indices into our field protocols? What will you...
Well, I think that I heard a lot of presentation where people were not at ease with their results because there were a lot of anthropophonic sources that were polluting their results. Well, it's a great way, as you heard with the stream completely not audible after spatial filtering. You could just take the information that you are interested in. So if you want to hear a specific species in this direction, you can do it. You won't have to bring a directive microphone and just do the recording just at the moment you need to. You can just bring your spherical microphone array and then extract only the signal in one direction. So it could be less of a stress for you and really useful, I think.
And when there are several bird species, it works too?
It works too,
right?
Actually, because they do not, like if you find two birds exactly in the same, with the same acoustic signature and same duration, it might not work, but I've tested it on bird choruses and it works well, so.
Thank you. Yes.
Hello, thank you for your presentation. I have a question, maybe I don't understood it very well. I didn't understand very well, but so the NRI is a tool for isolate some sounds, or it's a tool for calculate an indices of I don't know diversity of sounds or something like that?
Well, that's a very good question because initially it was developed by Sylvain Aupert to calculate like the state, like to correlate it with the state of biodiversity. So basically what they did is that they counted every acoustic event on the spectrogram. So every high frequency, low frequency, every event. And there was a big correlation with the state of biodiversity, so they were really happy about it. But us, we use it more to isolate just one species, to localize one species. So, and you could do it if you know that in your environment there are, I don't know, like 4 species of birds. You can do it 4 times to have the localization of the 4 species. So, yeah. But that's a great question because it's used in both ways by different scientists.
Okay, thank you.
We have the time for one more short question. Yes.
Hi, thanks for the talk. Did you have any? Did you do any tests on insects with this type of microphones? Here's the insect.
No, we didn't.
No.
But
Yeah, because I was just wondering, because one of the challenges about using passive acoustic monitoring to identify the species, it's exactly that sometimes it's hard to filter the noise or things like that. So I think I don't know, it might be like a useful tool for the insects as well, like to have this direction, especially because I don't know, they're so yeah, they like the vocalizations, they have like a very short range, and so if you really like have something to point at them, it might be, I don't know, it's try it, please.
yes, yes, that's a great idea.
Thank you very much, Maud.
I'm sorry, just for future speaker, try to speak right in the mic, because if we can hear you in the room, people online won't hear you if you don't speak. Like, they don't are very sensitive, so try to speak right in.
Hello. That's a perfect time to my throat, Jesus. Nice to meet you if I haven't met you already. I'm Ruari. I feel like this is going to be a really good follow-on from Maud's, because I'm basically talking about how noise is really bad if you're trying to detect any kind of species. So this might be like a nice ad for getting a spherical mic. I'm talking about some work I'm doing in my PhD. I'll be writing up soon, so don't steal it, maybe. But basically, if we would like to have abundance metrics from passive acoustic monitoring, that would be really nice. Right now, we can get pretty good abundance metrics from physical surveys. So this across the UK, we have a big breeding bird survey where we measure the abundance of like 100 and something species of birds. But ideally, we would like it on a finer scale to answer things like, how does this habitat type impact abundance? How does this type of land management impact abundance? Maybe you want to be able to quickly detect when your abundance declines, if there's like an invasive species or disease or something like that. But right now we can't do that. And one of the reasons is that, so the detectability of the species that you're interested in is really important. So these guys talked about the difference between absolute and relative abundance. So if you've got a community of, I'm talking about birds, but this can be related to any kind of species. A community of birds, if you have different detectabilities for your different birds, so red is undetected, black is detected. In this case, the little birds are not detected that well. The capercaillie type birds are detected quite well. You will end up getting a measure of abundance that doesn't look like exactly what the truth is. So it's really useful to know what the detectability is. And so this critically changes, or you would expect it to change, for example, by time. If your detectability increases over time, but your true abundance is decreasing over time, then the actual trend is that the abundance is decreasing, but you might not see that at all if you don't account for the fact that the detectability is changing. Similarly, this one was maybe more obvious. Across different habitat types, the detectability is going to change. So if you're in an open field, it's easier to detect the species you're interested in. If you're in a forest, it's going to be harder. So if, for example, you have more abundance in the forest than compared to the open field, but the detectability is decreased in the forest, then you might not actually see as well the benefit of having a forest on the abundance of the species that you're interested in. So that's quite bad. So it's quite important to measure the detectability and have like an idea of how it's changing. The problem is, for acoustic detectability at least, it's quite hard to measure, or it's annoying to measure. So one thing, this is 5 minutes of audio from my data set. It's a data set of about, in this paper, 2,000 hours, so it's not too huge, 28 sites. There's a lot going on here. So if you want to measure the acoustic detectability manually, you have to say, as a human, go through this and kind of pick out, well, first you would run your model on this to get detections, and then pick out as a human where the detections were missed, where the vocalizations were missed. So this is 5 minutes of audio. If you wanted to manually validate, I think I put 1% of my 2,000 hour data set. This is 5 minutes. 1% would be about the size of like 3 tennis courts of just spectrograms that you'd have to go through manually. So probably not not going to be very fun to do, and it takes forever. So yes, it's hard to do manually to measure the detectability. And as someone mentioned, Halper, they did this study where they found basically one of the key components of how easy it is to detect a bird is essentially the background noise that's happening. So if you've got, in this study, they had a bunch of, I think it was cicadas happening, and when those happen, you can detect a bird, or you can basically the detection distance goes from 50 meters down to like 2. So it's basically you can't detect anything if you've got background noise going on. So hopefully I've convinced you that the detectability is important if you want to measure abundance well. It's quite annoying to measure because it takes ages. And also background noise is a key component of the detectability. And with those 3 things in mind, I wanted to try and find an automated way to figure out how much the background noise is going to impact your detectability. And I'm going to walk through that now. So to start with, this might be a bit complicated, but to start with, essentially you run BirdNET or whatever model you're interested in on your audio data set, get detections, get confidence values. And in this case, you start, so the starting step to get foreground vocalizations, which is high confidence vocalizations that you would like to be able to reasonably detect in your data set. So here I picked 15 different vocalizations for each species that have above 0.98 confidence, most of them like 0.995, very, very high. This is for tree pipit, but I've done it, at least in this study, for 6 other species. I'm only going to talk about tree pipit and cuckoo. But essentially I picked different species that have either similar, like meadow pipit, tree pipit, quite similar, or quite different vocalizations, just to see how it makes a difference. So the next step, and this is the important step, I think, is per site. So for each of my 28 Scottish sites, I sampled randomly 1,000 essentially bits of background noise. And then what I do is I combine them with the 15 foreground vocalizations. So for each of the foreground, background combinations, so vocalization, background noise combinations, I randomly choose the foreground vocalization amplitude and combine them together to get some kind of augmented audio clip, which is basically, essentially what I'm saying is what would happen if a tree pipit vocalized at this amplitude at this time at this site, which is quite useful to know. And then from that, I will just pass that augmented audio clip through a model like BirdNET, get some output, which is confidence, the amplitude of the background noise, which I'm not going to talk about now, it'll be in the paper, and the amplitude of the foreground vocalization. And that's all a bit confusing, but I'm going to show some graphs which hopefully make sense. So this is a nice line. X-axis here is the vocalization amplitude of the species for a meadow pipit. Y-axis is the detectability at a confidence threshold. So 0.9, that essentially means the chance of detecting a vocalization above a 0.9 confidence. And I've done this for other confidence values as well, but obviously no time. So for example, how to read this, a minus 30 decibel vocalization at this site, site E of a meadow pipit, you would expect to detect it like 80% of the time-ish. A very quiet vocalization, this is like almost below the noise floor of the microphone, you would never expect to detect it, etc., keeps going. So this is on its own is like kind of interesting, but when you start adding other sites is where it gets more interesting. Oh, by the way, this is vaguely what this looks like. 1 minute of noise from this site looks like this. When you add other sites, it gets more confusing. So for example, at this site, the detectability is awful, generally. It's far, far worse. At this site, the detectability is slightly different. Definitely easier to detect at this site, site B. Site B is basically characterized by wind noise. And this site, site A, was characterized by lots of other bird noise happening at the same time. And then we also computed this for a bunch of other sites. But basically, the key takeaway is it's far harder to detect birds at some sites than other sites. And to make it more confusing, oh, by the way, it's also different for different species. So, for example, this site, Site E, is easier to detect cuckoos, but it's harder to detect metapipits. And it also varies temporally. So you can have, this is Site B again, at some point really high detectability in the survey, a week later really low, a week later really high again. Kind of annoying. This is due to basically a big storm front coming in. So lots of wind came in. And this also changes for different sites again. It's really complicated. And it changes again for different species. So this site, Site E again, decreasing detectability over time for metapipit, increasing detectability over time for cuckoo. Very confusing. But, so fortunately, essentially we can, our method kind of allows us to see and put a number on how much the detectability changes, which is quite useful because it changes a lot. But yes, we expect it to vary spatially from different sites and also temporally over the course of a survey. And hopefully, if, for example, you measure a bunch of detections in a period where there's low detectability, you can use that information on how detectable the species was at that time to give you a better metric of what your abundance actually truly was, like a more absolute measure. So yes, that's all I'm going to talk about. If you're doing anything to do with machine learning or abundance or, I mean, broadly birds, please talk to me, and I'll take any questions now. Thank you.
Thank you for your nice talk. I'm sure there will be a lot of questions.
Thank you. It was super interesting. I have two questions. I mean, I have one question is, did you publish this and can we read it somewhere?
Soon and soon.
That's
and
great.
I'm also going to be publishing the code and making it. It should be ideally so that you can kind of just press play and it goes to some degree. That's what I would like to do.
Okay, that would be great. I have more of a methodological question. So I want to follow a species for a year and be interested in their phenology. But how do I know the difference between my ability to detect them and actual phenology and actual abundance and actual presence of the species?
That's a great question. I'm not sure I know how to answer. There's a lot of different things that go into detectability. It is not just the background noise. So you could actually be measuring differences across the year in phenology or whatever. But this allows you at least to account for and reduce any biases that you would have if there was big changes in the background noise, for example, over the course of the year. Does that make sense?
I mean, I'll come talk to you at the end.
Yes, please.
Thank you so much.
I'm not sure I understood how you calculated your detectability. Would you mind going over it again, please?
Yep. So essentially, as in which part did you not understand? I'll go back to the slide with this is the last slides. This guy.
Oh yeah.
This part is essentially what I do is when I create my augmented vocalizations, run them through BirdNET, get a confidence score for a ton of vocalizations. So it's 15,000 different vocalization background clip combos for each site. And then for each of those combinations, I measure the confidence score and see if it's above or below a confidence threshold. So typically when you're measuring abundance, you'll count the number of vocalizations above a certain threshold, like 0.1, 0.3, 0.5, 0.9. For the graphs I showed, I was using a 0.9 threshold. And I essentially just, the percentage of these 15,000 vocalizations that were below a 0.9 confidence after you'd added the background noise. So they all start on like 0.99 something. And then looking at the percentage afterwards, after adding the noise, that are below a certain confidence. Does that make sense?
You might not have the answer, but I was wondering if how much this noise would also bother an observer who would be listening, and if it's worse for BirdNET or for a human. But you might not have the answer. That's okay.
The answer is it depends. Sometimes it's easier for humans, sometimes it's easier for BirdNET.
Okay.
I've definitely had some situations where it might be really noisy and an observer can't pick something up, but the BirdNET or whatever model can pick it out with spectrogram. As well as there can be situations where, if, for example, this vocalization is really easy, sometimes if you just put a big chunk of noise here, even if it has nothing to do with the vocalization, doesn't mask it or anything, it can still confuse BirdNET in a way that would not confuse a human. So it depends, basically, both.
Thank you.
Just a quick question to let time for other questions. Did you manage some temporary background noises, like road traffic or people talking away, and continuous background noises like electronic sounds or just microphone sensitivity or something like that?
The only thing I specifically tested, I tested two things, which was, well, I tested wind gusts and stuff like that against bird noise, which was continuous, and against just white noise, which I guess kind of sounds like road traffic or microphone noise. The worst one by far was bird noise, which kind of makes sense. If you've got other birds, it's harder to tell, like from a spectrogram, which is which. Wind was the easiest, because often, if you have a big blast of wind, if it doesn't cover the bit that BirdNET is interested in or needs to make a detection, then it doesn't matter. White noise was not too bad. But yes, so it depends on the type of noise a lot as to how much it's going to impact the detectability.
Okay, thank you.
We don't have the time for any other question, but you can talk with each other during the break, and we will listen to the next presentation. So well done.
Thank you.
Please welcome Maëlle Tisserant for her talk, the fourth of the morning, the last of the morning.
So hello, my name is Maëlle Tisserant, and this presentation will focus on enhancing automatic detection and identification of small mammals through sound. This presentation is part of my master's degree and based on my internship. My supervisor are Yves and Tiffen Deveau, and we work in collaboration with Stuart Newson from the British Trust for Ornithology. So first, you may ask why study the sound of small mammals. First, I will define what is a small mammal for this presentation. So for me, a small mammal is a terrestrial mammal weighing less than a muskrat. And we find rodent and insectivores groups. So for example, we have the hazeldarm mouse, hamsters, voles, and rats among the rodents, and among the insectivores we find the shrews, musaraigne in French, and the moles with, for example, the European mole. Then, why study these very diverse groups? So first, because they are of a great ecological importance. They are killing in food webs. They can be seed and they can play a role in speed and sport dispersal, and also they can be soil engineers and indicators of habitat quality. There are also many challenges regarding species conservation for these species. In France, there are 3 species with a protection status. They are protected by law, and they also can be vector of zoonotic disease or invasive species, especially in Iceland ecosystems. Also, there are some knowledge gaps regarding this, the study of these species. For example, a lot of species are data deficient on the IUCN Red List, and also we know very little about the species distribution and the dynamics of the population and of their abundance. So also, why study the sound of these species? So first, because the traditional methods for the study of small mammals are live trapping, camera trapping, the search for indirect evidences, like nest, species, burrows, feeding traces, the search for predation evidences, like pellets, feces, and the use of tracking and sampling tubes, and also environmental DNA. However, all of these techniques have limitations. They can be very invasive, like live trapping. They are labor intensive. They also don't always allow reliable species identification. It's a case for camera trapping, for example. And they are highly condition dependent, for example, the search for nests. And they have low detection probability. There is also a need for new methods, especially from wildlife managers, for time-efficient methods and also methods that are non-disruptive to wildlife. So acoustic can be a good solution because this method, as you know, is very time-efficient. It's non-specific, it's a passive and non-invasive method, and it is cost-effective in the long term. It is also suitable for hard-to-access environment and secretive species, so it's great to study small mammals. So now, how to identify a small mammal using sound? In the last 4 years, new knowledge has cleared up the ground for small mammal sounds study with the publication of a book and a few articles from Stuart Newson, Nae Middleton, and Yuma Pierce. And they published the first recordings and the first comparisons for most mammal species. And they also published the reference sound database of small mammal vocalizations. They have shown that most mammal species produce ultrasonic or very high frequency sound. So I'll show an example of the water shrew, Neomis phodians. This species is common in France, but it is also protected, and it emits sounds usually around 9 to 13 kilohertz, and it sounds melancholic. And when we listen to the sounds, there is no modulations in the calls. However, there are some data deficiencies in the sound knowledge of small mammals. Actually, acoustic data are lacking for 50% of small mammal species in mainland France, and the only species for which we have a good vocalization knowledge level are the house mouse and the rat species, because they are lab species or because they are common species. So now, where can small mammals sounds be found? So for my internship, I used a large sound dataset already existing from Vigikiro. Vigikiro is a French national bat monitoring scheme. It's a citizen science-based program consisting of standardized passive ultrasonic recordings. So it's suitable for studying small mammal sounds. And today it has accumulated more than 100,000 nights of recording and more than 25,000 sample points for, in total, more than 750 terabytes of data. And in this program, we have recordings. Then the recordings are run through a sound event detection algorithm, Talarida-D. This sound event detection algorithm is rule-based. And then the sound events are classified using a sound event classification algorithm, Talarida-C. This algorithm is a random forest classifier, and it uses a reference sound database of 80 classes. So first we have a lot of bat classes. There are species of bats. We also have Orthoptera, bird, human, and noise classes. And for the moment we have 4 classes of small mammals, but these classes are very imprecise. So we use a new reference database from Stuart Newson, consisting of 16 small mammal species. And the goal was to assess the performance of this new classifier of 16 new species. But the dataset is very large, very heavy, so we couldn't process the whole dataset. So we choose to select a few recordings. First, the first method we used was a random selection of recordings. We use a clean Vigikiro recordings and randomly selecting around 20 recordings. We run them through the sound event detection and sound event classification algorithms. Then we manually validated all the identifications. And the results we have were that there are a low diversity and a low detections on all the nights, with only 5 species that are present on the nights. The great white-tooth shrew, the rat species, the wood moose, and the crown and common shrews, because these species are ubiquitous, anthropophilic, and we did not find any rare species. It was like looking for a needle in a haystack, so we chose another method. We did a targeted selection of recordings based on 2 selection filters. The first filter is a species selection. We choose 3 species with non-vocalization that are easily distinguishable from other species and with a well known habitat. Then we did a second filter based on distribution and habitat selection. So I will show an example with the Hazeldown mouse. So for this species, we also used the clean VGKRO recordings, and we selected the recordings within the 10 by 10 kilometer grid cells where the species is already documented. And then we selected the recordings based on habitat suitability. So for example, we choose the recordings next to hedgerows, next to forage edges and bocage landscape because this is the habitat of the Hazeldown mouse. Then we randomly selected 50 sites inside this selection. Then we are planning to run the recordings through the same algorithms and to validate all the identification manually. We will add all the new identifications to the referencing database, and the results for this method are still to come. So now, what next? The goal of my internship was to do an overview of small mammal vocalization data in the VGKRO dataset, and also evaluating the performance classifier, the classifier performance, and I'm still doing that. We are planning to do a comparison with other algorithms, for example, like CNN. But there are also some other research gaps and open questions on small mammal studies. For a lot of studies, species, as I said, there are unknown vocalizations. Also, we don't know about the species detection rates and the detection phenology. Also, there is a need to compare acoustics with other survey methods and to know more about the intraspecific variations of the vocalization of a species and the interspecific differences. So if you are studying acoustic recordings, please consider small mammal sounds during acoustic analysis, especially if you're working on ultrasonic data. And thank you for your attention. If you have any question, please feel free to ask now or later.
Thank you, Maëlle. Nice presentation. Do you have any question for Maëlle? Come on. No? Maybe I have one. What adaptations were needed to use Tadarida on small mammals, because it was based on bats, not small mammals. So did you encounter specific difficulties or mismatches or whatever?
The biggest challenge was to have a good reference and database, because apart from the work of Stuart Newton, Hamilton and new mappers, there is no reference and database of small mammals. So the first things we did was to try to construct a reference and database of small mammals, but it was very hard because it was hard to find any small mammal sounds in the VGKRO datasets. So that's why we use the sound database of Stuart Newton. So now this challenge is overcome, but yes, I think this was the main issue we have with using Tadarida for small mammals. And also because we need to process a lot of data that are already processed in the pipeline of VGKRO, and we have to do another round of processing. So it takes a lot of time. So I think this is also another big challenge.
Hi, thank you, great presentation. I wanted to know how you got the idea of a bat dataset to look for small mammal sounds.
Can you repeat your question?
Yes, of course. How did you get the idea of using a bat dataset, so the VGKRO dataset, to look for small mammal sounds?
It was actually not my idea. It was the idea of my supervisor, Yves. But I think he read about the work of Stuart Newton and heard about it, because a lot of small mammal species, as I said, produced ultrasonic vocalizations. For example, it can be higher than 50 kilohertz. So I think Yves wondered if this vocalization could be found in the datasets, because the dataset is made of ultrasonic recordings. So I think that's where the idea is coming from.
Hey, it's me again. Thank you very much for the presentation. I was wondering what type of feature does Vigikiro Tadarida or Tadarida C also takes as like input in the random forest?
There is plenty of features. I don't know all the details, but it's mainly frequency features, also intensity and duration of the calls. I don't know if there are also all the parameters that were presented on the presentation before, but yeah, maybe I can answer in more details after. I just have to look at the methods and yeah.
Okay. Thank you very much, Maëlle.